Are you prepared to testify under oath here today that COVID-19 is a bioweapon?
Yes, I am.
Dr. Fleming, we've asked you to come testify about whether COVID-19 and related vaccines are a deliberate bioweapon attack on the United States. Madam Court Reporter, can you please identify yourself for the record?
My name is Karen Usher, Texas CSR number 5536.
Thank you. Can you please swear in the witness?
Dr. Fleming, will you raise your right hand? Do you swear the testimony you give today will be the truth, the whole truth, and nothing but the truth, so help you God?
I do.
Dr. Fleming, do you consent to this testimony being used in any appropriate proceeding?
Yes, I do.
First, I'd like to cover some of your qualifications. You have an impressive 90-page resume. I'm not going to go through that, but very briefly, are you a medical doctor?
Yes, I am.
So therefore, you have an MD degree, as it's known.
I do, yes.
And what is your area of specialty?
Nuclear cardiology, and internal medicine.
And do you also belong to professional medical associations?
Yes, I do.
Are you a fellow of the American College of Physicians?
Yes.
And are you a fellow of the American Society of Internal Medicine?
Yes.
And you also hold a PhD?
Yes, I do.
And in what field?
Physics.
And you also have a law degree, otherwise known as a JD?
I do.
And are you an inventor in the medical field?
Yes.
And very briefly, can you explain the type of medical inventions that you've invented?
The ability to image the body and determine what types of disease processes are going on by looking at metabolism and blood flow.
And do you hold patents for your inventions?
Yes, I do.
Is it fair to say you're familiar with the world of medical patents?
Yes.
You have the ability to read and interpret medical patents?
Yes, I do.
And are you also an author of professional papers?
Yes, sir.
And approximately how many?
Somewhere between 400 and 500.
And you also serve on the editorial board of professional journals?
On editorial boards and review boards for the journals.
And briefly, what does a reviewer in a professional journal do?
The obligation is when somebody submits a paper for publication for reviewers to take a look at that material, to determine if it's scientifically valid, to determine if it makes a difference in the field, and to make recommendations back to the editorial board about whether the paper should be accepted or revised or rejected for publication.
Are you prepared to testify under oath here today that COVID-19 is a bioweapon?
Yes, I am.
You wrote a book posing the question, is COVID-19 a bioweapon? Correct?
Correct. The function of the book was to provide information that the general public could look at, and also to provide evidence for the legal system and the medical system to understand the gain-of-function research that's been carried out over a couple decades, to show where the monies came from, to show what papers were published, showing what types of gain-of-function research occurred, and to show the patents, to clearly lay out 2 decades worth of work that's been conducted, funded primarily by the United States, although other countries have been involved, and to show how there's no evidence that SARS-CoV-2, the virus that causes COVID-19, no evidence to show that it's naturally occurring, no animal model for it, and to show the type of research that's been done with coronaviruses, funded by the U.S., supported by NIAID, NIH, Department of Defense, monies that went to Peter Daszak at EcoHealth, to Ralph Burrick at the University of North Carolina, to Shizheng Li at the Wuhan Institute of Virology, and to several other universities around the country, including in the state of Texas.
Did you also look at the genetic code of the SARS-CoV-2 virus, and especially the spike protein?
We did, as well as Professor Luc Montagnier, who is the gentleman who discovered the HIV virus, and several other individuals that we note in the book.
And based on that review of the genetic code, that informed your conclusion that this is a bioweapon?
Right. So the Biological Weapons Convention Treaty states that any adjustment or modification of a biological agent like this virus that doesn't provide a benefit for mankind is a biological weapon. These particular changes in this virus, including the PRRA insert, which are amino acids that were inserted that are very critical for the furin cleavage site for this virus to infect. The insertion sites that were made with HIV and simian or ape-like HIV equivalent viruses, the prion-like domain at the top of the spike protein, as well as the HIV glycoprotein 120 insert that Shi Zhang Li put in early on that's critical for the attachment of this virus to cells, all of which are not naturally occurring.
So to be clear, the SARS-CoV-2 virus, otherwise known as COVID-19, and especially its spike protein, is not something that's simply involved on its own in nature.
We've looked at all the different coronaviruses that exist on the planet. None of them have the PRRA insert. None of them have this tremendous amount of HIV insert. None of them have a regional binding domain that is a prion-like binding domain, which means that where it attaches to the cells, it's a prion-like domain, and prions are things that are abnormal proteins that cause other proteins to become abnormally folded.
So to be clear, genetic changes were made to the spike protein in a lab to create what is known as the COVID-19, correct?
That's what all the data shows.
Your book references research in 2010 by Shi Zhang Li, who's often called the Bat Lady. Was that research designed to determine how to increase the spike protein binding capacity to the human ACE2 receptor?
So that was her goal of that research, was to increase the ability of the virus to infect human cells.
Now, interestingly, I think in this published research, they discovered that the naturally occurring spike protein from horseshoe bats was unable to bind to the human ACE2 receptors.
Right, that's exactly right.
So as of 2010, scientists, including Shi Zhang Li in Wuhan, China, knew that naturally occurring horseshoe bat coronavirus spike proteins could not infect humans.
That's right.
Based on your testimony, it sounds like you discovered at least 2 man-made inserts into the SARS-CoV-2 spike protein genetic code that prove it was produced in a lab.
Right. The research papers that Shi Zhang Li and Barrick and others published, they were very clear and somewhat proud of the fact that they'd been able to modify the virus and to insert changes into it that made it more effective and potentially more dangerous.
Is there some laboratory mechanism where they're able to take genetic code from elsewhere and put it into a virus?
Right, so you can actually build anything if you know what the structures are. It's like building a car. If you know what the parts are, you simply put it together. Many of the research studies that we have pointed out, they took bits and pieces of one virus, combined it with bits and pieces of another virus. If you go and look at those papers, you'll also find that they were very specific by inserting 5 nucleotide base pairs in there. So they simply switched the base pairs, and that will result in a different amino acid being added and a different protein.
Let's talk about the first insert that you found in the SARS-CoV-2 virus that led you to conclude that it was a man-made bioweapon. The first one I think you referred to is the furin cleavage PRRA insert, correct?
That's correct.
And can you explain briefly what that is?
Okay, so that's 4 amino acids that were put in. When this virus attaches, and I know this is long and complicated because a lot of people have heard of ACE2, which is one of the receptors, but it's a multi-step process. The HIV glycoprotein 120 insert, which was the second one we talked about, is required to attach to what's called a sialic acid receptor raft that stabilizes it and puts it in position. The virus then comes into contact with the ACE2 receptor. That then undergoes a change with something called a TMPRSS2 receptor, which is critical to understanding because that begins the process of bringing that virus into the cell. It then has to undergo furin cleavage at the furin cleavage site, and that's the PRRA, which makes it particularly infective for human cells. And then it comes across also something known as the neuropilin-1 receptor, which is found in the brain.
How did you discover that this PRRA furin cleavage insert had been made to SARS-CoV-2?
All you have to do is the nucleotide-based sequencing, which is how we know all the variants occur. And when you do that, you find this PRRA insert.
Now, this genetic code for the furin cleavage insert, the PRRA, is that found in any other coronavirus in nature?
None.
It's not found in any other coronavirus that exists in the wild?
Right. There's absolutely no other coronavirus that has this PRA insert. And that's 12 nucleotide bases, not a single base like sickle cell, but 12 specific nucleotide bases to provide for 4 amino acids that very specifically is connected with the furin cleavage site, which the US government owns the patent on.
So the fact that this insert and this genetic sequence for this insert is not found in any other coronavirus in nature, what did that tell you?
Well, since mutations occur one nucleotide base at a time, you would have to come up with some phenomenal explanation for why 12 nucleotide bases suddenly inserted themselves into a virus that none of the other viruses in that category have. The original premise that was put forward and actually published in The Lancet was that this was a naturally occurring virus with an animal host that got transmitted to people, but yet there's not been a single animal model found with this. Neither has there been a single coronavirus that has this PRA insert.
You mentioned the second insert, an HIV glycoprotein.
Right.
So are you saying that sections of the genetic code for HIV were inserted into the SARS-CoV-2 virus?
Yeah, Xi Zhengli published this early on, many years ago, where she was very proud of the fact that she did this. She used an HIV pseudovirus to do it. And we also know that HIV glycoprotein 120 is a prion.
So this is the HIV that became famous in the 80s and 90s. It's that HIV.
Right.
And somebody took pieces of the genetic code of HIV and put it into SARS-CoV-2?
That's what Xi Zhengli admitted to in publications that she had, yes.
Were you able to discover why that was done?
According to papers that she published, it was to increase the infectivity to see if she could make it more infective. But it's even clearer that what she was going for was the development of a prion disease, because where everybody is kind of coming up to speed with a virus and how challenging it can be to deal with, what's even more challenging to deal with are prions, because prion diseases are relatively new in our understanding for treatment, and they cause permanent damage unless they can be reversed. And so the development of a prion, putting HIV glycoprotein 120 in there, attaching to the sialic acid receptor, there's nothing about that is advantageous for people. And the ability to produce prion diseases is quite a weapon.
And how lethal are prion diseases?
People die from prion diseases.
Is this process of inserting these amino acids to increase the infectivity, is that gain of function?
Right. So gain of function is anything that you do that changes a naturally occurring biological agent like this virus. And so the premise of gain of function really has a good premise behind it. The original idea being that if you could stay a step ahead of a potential infectious agent, say a coronavirus, if you could stay just a step ahead, you could do a good job of providing treatments for people and knowing when and where it was going to spread. This isn't a step ahead. This isn't a single nucleotide base. This is a spike protein that has an HIV glycoprotein 120 inserted into it. This is a spike protein that has 4 amino acids to make a furin cleavage site, the PRA insert. This is a spike protein that has multiple other, according to Luc Montagnier, HIV and SIV inserts. This is not something that evolutionarily evolved. The evolution of something with this many changes would take hundreds or thousands of years. This is not staying a step ahead of it. And making this provided no advantage for human beings, which makes it a Biological Weapons Convention treaty violation. It's playing God.
To your knowledge, has anyone been able to disprove or refute the fact that there are artificial man-made insertions into the SARS-CoV-2 virus?
Actually, there's been no dispute of that, except for the statement that it's naturally occurring. And then when you say, well, where's the scientific evidence that it's naturally occurring? There's no follow-up to that. So that for people that are claiming that this is a scientific discussion, I would encourage them to stay with the fact that this is a scientific discussion. There's no science to show that any other coronavirus on the planet has these inserts. There's no science to show that there's a natural reservoir in animal for this virus to occur. In fact, the science points to the fact with over 99% probability, which is what we're dealing with until somebody comes up and confesses, that this is not naturally occurring.
And in your mind, is there any doubt that without this gain-of-function manipulation of the SARS-CoV-2 spike protein, there would have been no pandemic?
we wouldn't have had this virus not been released.
Have any of these folks, Dashak, Barrick, Xi Jinping, Fauci, explained what that beneficial alleged effect is?
Well, up until now, it seems like most of their conversation has been, we didn't do anything like that, even though the data is now showing it. But as we've learned over the last couple of months, the National Institutes of Health and National Institutes of Allergies and Infectious Disease have now admitted, not that they needed to, but it's nice for them to admit that a gain-of-function research was funded by the U.S. and that Fauci's statements that they weren't involved in gain-of-function research was not correct.